Okay. Well, hello, everyone, and welcome to our July edition of Science Cafe with the Conservation and Research Department here at the Atlanta Botanical Gardens. We're very happy to have with us today from Berry College, Dr. Caitlin Conn, who will be presenting on parasitic plants and the search for the host with the most. Just want to let folks know that we will be recording this event. And so if there are any folks that would like to listen to this at a later date, you'll be able to do that, or if you want to share it with friends. And I've gone ahead and shared in the chat a link to our website where you can check those out. And you can also check out future events and even some of our past events if you'd like to listen to those. Also, I want to let everyone know that at the bottom of your screen, there's a little question and answer feature, and y'all are welcome to direct any questions that you might have for Dr. Conn there. And then at the end of her talk, here in about 30 or 45 minutes or so, we'll go through some of those questions and try to get as many of those answered for y'all as we can. So again, just please direct those to the question and answer feature. And I'd also just like to let y'all know a little bit about what Dr. Conn has been up to these past, well, really her whole life, but here's a quick bio. Dr. Caitlin Conn grew up in central PA, where she developed an early love for nature on her family's farm and forest. As a biology major at Penn State, she conducted research on lizards from Caribbean islands and earned her Bachelor of Science degree in 2011. Next, she moved to the University of Georgia, where she studied the genetics of parasitic plants and earned a PhD in 2017. After 2 postdocs at Spelman College and Emory University, Caitlin joined the faculty at Berry College in the Department of Biology. And she runs a research lab there that focuses on local parasitic plants, such as false foxglove and bearcorn, and American chestnut trees. So without further ado, Dr. Conn, I give you the floor.
All right, thank you so much for the introduction. So I'll go ahead and share my screen with my slides, and you can just let me know if there are any issues. Let's see. All right, is everybody able to see my screen?
Looks good.
All right. Great. Well, we'll get started then. So thank you all so much for being here. Thank you to the Atlanta Botanical Garden for inviting me. This is only my 3rd ever invited talk as a professor, so I'm really glad that we could make it work virtually. I'm currently in my 2nd year as an assistant professor at Berry College, as you heard, and I'll talk more about Berry College in just a few minutes. So teaching is a big part of my job here, but I also run a small research program, and that's mostly what I'll talk to you about today. As you heard, it's about parasitic plants, and parasitic plants are plants that latch onto other plants and steal resources. So on the right side of this slide, you can see a parasitic plant right here. It's this tiny white seedling coming up out of the roots of a host plant. The parasite looks small relative to its host here, but it's capable of doing quite a bit of damage, and we'll see that shortly. So before we talk more about my current research, I want to quickly discuss how I got where I am today, and then we'll talk about my past and present research on parasitic plants. And at the end of the talk, we'll discuss the fact that parasitic plants aren't all bad. In fact, there are some pretty cool ones right here in Georgia that you might get a chance to see and admire one of these days if you haven't already. But I bet some of you will be familiar with them. So I went to Penn State as an undergrad, and then for grad school, I knew that I wanted to go on with something in the life sciences so that I could have a career focused on research. And what I really, really wanted to study was infectious diseases. I was really fascinated by the pathogen side of host pathogen interactions. And so that's what I thought I was going to do with my life. But I went to the University of Georgia for grad school, and I tried out a couple of different labs. And as I got more exposure to using animals in the lab, I started to realize that that wasn't for me. To this day, it's a struggle for me to kill a cockroach. So, infecting cute little mice with some kind of nasty pathogen was not going to work out for me. But I stumbled on a lab that allowed me to study parasites, so close enough to infectious diseases, without ever touching an animal because this lab studied parasitic plants. So, more on that in a minute. But really, this lab, led by Dave Nelson at UGA, was focused on signaling in plants. And in particular, we studied two really cool groups of signaling compounds. So the first group is represented over here. These compounds are called characins, and they're derived from burning plant material. They stimulate germination in some plant species after a fire, when other plants have presumably been eliminated. So, characins are essentially a signal that plants are absent, and that's probably a good time for most other plants to germinate because competitors for light and other resources have been eliminated. Now, the other group of chemical compounds are called strigolactones. They're over on the right side of the slide, and they are plant hormones. Since they're made by plants, they tend to be abundant in the environment when plants are also abundant. And strigolactones have all sorts of important functions in the development of the plants that make them. They also go out into the soil where they signal to beneficial arbuscular mycorrhizal fungi. And to parasitic plants, they're a cue that lots of other plants are around. So it's probably a good time to germinate and find a host. And we'll talk much more about them a little bit later. So after grad school, I did those two postdocs at schools in Atlanta, Spelman College and Emory University. And then I got this job as an assistant professor of biology at Berry College. And Berry is such a unique place. If you haven't been up to see the campus, I really encourage you to come up and check it out sometime. So the picture on this slide shows a few of the really beautiful buildings on one end of our campus. We have about 2,000 undergrads, very few grad students in a couple of different programs, and our class sizes are small. We have a pretty low student to faculty ratio, and Berry is really huge on mentorship of students. So the student professor relationship here is really close, and the students seem to like it that way. Another really amazing thing about Berry is that we have a 27,000 acre campus and most of it is forested. So if you come to check it out, bring your hiking shoes or your trail running shoes, bring your mountain bike because we've got trails all over campus and it is just so much fun and so beautiful to go out there and explore. And I am not a field biologist by training, so I don't have much experience working away from a laboratory out in nature, but I think I had better become a field biologist pretty quickly since most of the campus up here is really a giant field site. So that's enough about my background. Let's talk a little bit about science. So I'm going to introduce you to some of my favorite organisms momentarily, and you can probably guess that they're parasitic plants. But let's take a step back for a second. When most people hear the word parasite, we think of organisms that cause disease in humans, livestock, or pets. So some familiar examples are, for instance, Plasmodium falciparum, the parasite, the protist parasite that causes malaria. Salmonella, this is a bacterial parasite that's often implicated in foodborne illness. And yes, tapeworms, the parasites that almost every pet owner and farmer has to deal with at some point. And viruses can also be considered parasites, even though they don't exist as cells and aren't really considered alive. Right now, we're all very familiar with acellular parasites like the virus that causes COVID. But parasitism is quite simply a type of symbiotic relationship in which one organism benefits at the expense of the other. And it's found across the tree of life. And more plants are parasitic than many of us realize. So let's discuss a couple of noteworthy examples. Probably the most famous parasitic plant around here in Georgia is mistletoe, here on the top left. So, mistletoe parasitizes trees, and it's particularly easy to see in the winter when deciduous trees have lost their leaves and the parasitic mistletoe is visible as clusters of green up in its host's bare branches. So, mistletoe attacks tree hosts, as do some of the parasites that we'll talk about in more detail today. Here in the middle, we have one called corpse flower, and when it blooms, it produces the largest single flower of any plant species in the world. But prior to blooming, this parasitic plant spends its life inside the tissues of a host plant. And as you can see, the flower is really beautiful, but it smells like a corpse, and that attracts its pollinators. So that's where it gets its name. And on the right, you have one that you saw on my title slide. This parasite is called Striga hermonthica. And Striga species attack important grass crops, so they're very agriculturally destructive. They cause billions of dollars of damage every year in sub-Saharan Africa. Now, all of those parasites across the top of the slide are characterized by a haustorium, and that's an invasive organ that they use to get inside host tissues and steal resources. There are some other plants that are sometimes considered parasitic as well, and these are the mycoheterotrophs. So they don't steal from a host plant directly, but they steal from a fungus that is in a mutualistic relationship with another plant. And a fun fact about mycoheterotrophs is that many orchids are mycoheterotrophic, at least at some life stage. Now, true parasitism, in which a plant lives in or on a host using a haustorium, has arisen 12 to 13 times in flowering plants. So, in this figure, we see the different groups of plants in which parasitism has independently evolved. Some of the plants represented here are hemiparasites, which still photosynthesize, although sometimes pretty inefficiently. Others, called holoparasites, do not photosynthesize. So, holoparasites always need a host, and they're also known as obligate parasites. Some hemiparasites need a host if they're not photosynthesizing all that well, but others are facultative parasites that can live with or without a host. And again, the one unifying feature of all true parasitic plants is that specialized invasive structure called the haustorium. So the haustorium extends out from the parasite to break into the roots or shoots of a host. And after a parasite's haustorium connects with a host's vasculature, the parasite can begin to steal nutrients. For much of the remainder of my talk, we're going to focus on the Orobanchaceae family, circled up at the top of the figure. And the members of this family are root parasites, that's indicated by the R in parentheses here. Some other parasites in this figure are shoot parasites, indicated by an S. And we also have endophytes indicated by an E. And an example of an endophyte is that corpse flower that you just saw that spends most of its life inside the tissues of a host plant. So for the Orobanchaceae, as root parasites, they attach to host roots underground, and they often begin to damage their hosts before they ever become visible above the soil surface. Parasitic plants from this family, the Orobanchaceae, are found all around the world, and many of them are what we call non-weedy. And this means that they occur in natural ecosystems where they parasitize wild hosts. The non-weedy parasites often have relatively narrow host ranges, and a lot of times they do little noticeable harm to their host plants. So we have some examples here. This is one that some of you may be familiar with. It's called Canophilus americana, one common name is American cancer root. It completely lacks chlorophyll and it kind of looks like pine cones sticking up through the soil surface. So you may have seen it in the forests of Georgia or really up throughout the eastern United States. And this tiny little plant, Canophilus americana, parasitizes oak trees, and the oaks really don't seem to suffer that much from it. Now, down here, we have another parasitic plant that's very different from the Orobanchaceae. This one with the white flowers and lots of green in the picture is one called Triphosaria versicolor. So this is a facultative hemiparasite. It can photosynthesize just fine on its own, but it will use a host if one becomes available. Now, on the right side of this slide, we have weedy parasites in the family. And weedy parasites are so named because they attack plants that are cultivated by humans, including lots of important crops. The weedy parasites tend to be generalists, so they can often attack a wide variety of important crop species, and they cause a lot of trouble for farmers. And one reason for this is that they germinate underground and they very quickly latch onto a host's roots, and they often cause considerable damage before they even emerge through the soil surface. So by the time they're visible, it can be too late to save the crop yield. So down here in this picture, we have a tobacco plant with green on it. And right down at its base, we have a tiny little parasitic plant called Philippanche ramosa. It has kind of tan stems and whitish purplish leaves. So parasites in the genus Philippanche and the closely related genus Orobanchaceae can parasitize a wide range of important crops, including, of course, tobacco and also sunflowers, tomatoes, carrots, and legumes. And then on the bottom right, we have a field infested with Striga hermonthica, also known as witchweed. Striga species are especially problematic in sub-Saharan Africa, where they cause billions of dollars of yield losses every year. So some parasitic plants in this family are very destructive for farmers. So here we have a simplified phylogeny, or a tree-like structure showing the evolutionary history of representative members of the Orobanchaceae. And what you can see in this figure is that we have a broad range of host dependence in this family. There's a progression from no parasitism. That's the case for Lindenbergia philippensis. It can't use a host because it's not parasitic. Then we have those facultative hemiparasites like Triphosaria versicolor, can use a host, can get by on its own. Then we have an obligate hemiparasite, Striga hermonthica, which can photosynthesize but can't get through its life cycle without a host. And then all the way on the right, we have an obligate holoparasite with no chlorophyll. And an example of that is Philippanche aegyptiaca. So we see that the obligate parasites absolutely require a host in order to survive, and the really destructive parasitic weeds tend to be obligate parasites. So their situation in life is really best summarized by lyrics to a song from Aladdin. They've got to eat to live, and they've got to steal to eat. Hopefully everybody here remembers Aladdin. So part of the reason why they have to have a host to get through life is they produce thousands of very tiny seeds, which don't have enough nutrient reserves to power a seedling for more than a few days after germination. So in this picture down here, you can compare the size of Striga seeds right here relative to the seeds of hosts, sorghum, and maize. These Striga seeds really just look like tiny specks of dust. So if these obligate parasites are so helpless on their own, how do they ever survive to find a host? Well, it turns out that many obligate parasites have an adaptation that helps them maximize their chances of finding a host. Specifically, they have to perceive host hormones called strigalactones in order to germinate. So here we have parasite seeds in the soil in this little cartoon, and they can remain dormant in that soil for years or even decades. And they're only going to germinate when they perceive a nearby potential host. So here's our potential host, and it's sending strigolactones in the soil to communicate with beneficial arbuscular mycorrhizal fungi. But the seeds of this parasitic plant are eavesdropping on that communication with arbuscular mycorrhizal fungi. They sense that a potential host is nearby, and only then do they germinate. They quickly attach to that host plant and they begin to damage it pretty instantly. Strigolactones also play lots of other roles in non parasitic plants, but we'll mostly focus on the fact that they stimulate germination in parasites today. So, an important thing to note here is that understanding how parasites perceive their hosts could help lead to new parasite control strategies. As I mentioned before, parasites begin damaging their hosts before they're visible. So, hand weeding, for example, is not an effective control strategy. But if we could manipulate parasite seed germination, we might be able to keep them from reaching their hosts in the first place. But to learn more about how parasitic plants perceive their hosts, we had to take a broader view of strigolactones and other similar compounds and how their signals are transduced in plants that we know a little bit more about. So, we had a couple of genes in mind that we thought might have evolved a role in strigolactone responsive seed germination in parasitic weeds. And one of those genes that stood out to us is called Ki2. When we started out, we had no idea what it did in parasitic plants, but we knew that it was involved in seed germination in a model non parasitic plant called Arabidopsis thaliana. Now, as far as we know, neither Arabidopsis nor any other non-parasitic plant can germinate in response to strigolactones. But in Arabidopsis, Ki2 does respond to compounds with some structural similarity to strigolactones. So this was a promising candidate for us. So we looked for Ki2 sequences in parasitic weeds, and we found out that they have a lot of copies of the Ki2 gene. So the Ki2 gene has undergone duplications in parasitic plants. And that was interesting for us because when extra copies of a gene are present, there's a possibility that some of those extra copies have evolved new functions. That's called neofunctionalization. And it's more common in duplicate genes than in single copy genes because when you have one copy of an important gene, that single copy probably can't change too much because it's got to fulfill its original function. If it changes to pick up a new function, that could be really catastrophic because its original function can get lost. But when extra copies of a gene are present, as long as one still has its original function, others can be freer to diverge. So there are a couple cool examples of duplicate gene evolution in action from other species. I think the coolest one comes from Antarctic fish, where duplication of glycoprotein genes enables them to survive in really cold waters. In E. coli, evolution of duplicate genes has been linked to heat tolerance. And in cattle, evolution of duplicate genes has helped them adapt to domestication. So in our case, we were really interested in the possibility that some of these extra Ki2 gene copies had undergone neofunctionalization in parasitic plants. So if at this point you're thinking that this sounds like a really cool question to address, I agree. I think it is too. And I hope you won't be too disappointed when I tell you that I've never worked with parasitic weeds directly. Because of the agricultural damage that they can cause, it's really difficult to get permission to work on the weedy parasites in a lab in the United States. So, to figure out what parasite Ki2 genes do, we had to take a different approach. We had to get those genes out of the DNA of parasitic weeds, And we had to put them into our model non-parasitic plant, Arabidopsis. So this is Arabidopsis right here. Some of you might be quite familiar with it, and it's really a model organism. It's a model organism for plants because it's easy to grow, it has a short generation time, it produces lots of offspring. We know lots about its genes and what they do, and it's pretty easy to take genes from another organism and put them into Arabidopsis. So that's why it's a model. That's why we wanted to work with it. So I put parasite Chi2 genes into Arabidopsis and ended up with transgenic lines, which are those Arabidopsis lines carrying Chi2 genes from parasitic weed species. So maybe I lost cool points for not working on parasitic weeds directly, but I did get to make frankenplants in a safe, controlled lab environment, and that was pretty cool too. Now, with these transgenic lines, I looked at seed germination with and without stragolactone treatment and asked whether those parasite Chi2 transgenes enabled a germination response to stragolactones, which is not something that we would normally see in Arabidopsis. So here are the results from a germination test in which we looked at our transgenic lines carrying parasite Chi2 genes. So here we're looking at percent of seeds on a plate that had germinated at a particular time point. So first, let's look at germination of plain old Arabidopsis with no transgene. So over here, the white bar represents germination on control media, so no stragolactones added in. The blue and purple bars represent germination on media supplemented with two different stragolactone mimics, one in dark blue, the other in purple. And you can see that Arabidopsis has germinated to some extent on its own, but there is no boost in germination with stragolactone treatment. And that is exactly what we would expect to see for plain old Arabidopsis. Now, here are the results for three different transgenic lines, each carrying a different Chi2 gene from parasitic weeds. And each Chi2 gene from a parasitic weed enabled a positive germination response to both stragolactone mimics. This was a really exciting result because it indicated to us that, yes, some Chi2 genes in parasitic plants likely enable host-responsive seed germination, just as we hypothesized. Other research groups have further characterized parasite Chi2 genes in the time since we published this work, and their conclusions are similar. Some Chi2 duplicates in parasites respond to stragolactones. So that's the big takeaway from our discussion of my past work in graduate school on parasitic plants. They use genes called Chi2 to sense chemicals coming from hosts and to germinate in response. So now we fast forward to the present, in which I run a small research lab at Berry College. And I told you earlier that parasitic weeds are really difficult to study in the US because they're so dangerous and their seeds are so tightly regulated. But fortunately for my research team and me, there are non-weedy parasites all around us. So in my current research program, we're focused on facultative hemiparasites, which photosynthesize just fine, don't require a host to survive, and usually aren't harmful to agriculture. Because they don't attack farmers' crops, they're not as critical to study, and a lot isn't known about them. But from an evolutionary standpoint, they are fascinating. And in particular, we're interested in how they have evolved and what genes allow them to detect and interact with hosts. So one question we're asking is whether facultative hemiparasites that can use multiple different hosts prefer one host over another. And really, how important is it that they find a host at all if they can photosynthesize on their own? My research student, Rachel Christopher, on the right in this picture, she wouldn't turn to face me for a photo, but there she is. Rachel Christopher was addressing this question with parasitic plants that use oak trees as hosts. Now, these parasites are different from Canophilus americana, which you saw before. These are facultative hemiparasites that are easy to grow in the lab. So she took 3 different parasite species and planted them with no host, a water oak host, or a willow oak host. So 2 different species of oaks. And then after the parasites and hosts grew together for several months, she measured the largest leaf on each parasite. Now, this was very much a preliminary experiment. It got a little messy at times. We had a small sample size, so we didn't run any statistics on it, and we definitely need to repeat it and expand it out in the future to draw conclusions from it. But in this graph, we're looking at the area of the largest leaf on parasites from 3 different species when they're with no host, a water oak, or a willow oak. So one thing that stood out was that when the parasites grew with no host, she observed pretty high mortality, at least for the 2 Areolaria species. Now, the Areolaria species in medium and dark green seem to do much better when they had a host than when they didn't have a host. You can see that their leaves were larger on average in the presence of a host, indicating faster growth. The pedicularis species in light green didn't really seem to do much better with a host than without, if it was able to survive without one. So again, our sample size for this experiment was quite small. We'll want to repeat it in the future, and we'll want to look at lots of other oak species as potential hosts to see if there really are preferences for one host species over another among these facultative hemiparasites. But so far, this experiment looks somewhat promising in terms of getting an answer to our question of host preference in some of these parasites. Now, here's a second question that gets a little bit more complicated. Remember that in grad school, my team and I found that parasitic weeds, which require strigolactones to germinate, perceive those strigolactones with some of their many copies of the Chi2 gene. Now, there's a non-weedy facultative hemiparasite called Trifasauria versicolor. You've heard of it earlier in this talk, and it does not need strigolactones for germination. But it does have lots of copies of that Chi2 gene, and some of them have evolved pretty similarly to the ones that encode strigolactone receptors in other parasite species. So my student Sarah Quigley, down here in the right-hand corner, is addressing the question of what those extra Chi2 genes do in Trifasauria if they're not required for germination. Conveniently, and in contrast to the parasitic plants that I studied in grad school, this is another one that it is safe and easy to grow in a lab. So one of Sarah's experiments investigated whether, even though Trifasauria doesn't need strigolactones to germinate, it might boost its germination rate when it senses those host hormones. So she put Trifasauria seeds on plates, and those plates contain different concentrations of strigolactones. So from left to right on this slide, the treatments were no strigolactone right here, smallest concentration of strigolactone, intermediate concentration of strigolactone, and highest concentration on the right. And as you can see, there's really no difference in the germination rate of Trifasauria, regardless of how much strigolactone you give them. So in this experiment, it looks like Trifasauria isn't using those extra Chi2 genes to perceive strigolactones at the seed germination stage. But why doesn't Trifasauria have a positive germination response to strigolactones? It could just be that the proteins encoded by Trifasauria's Chi2 genes are indeed incapable of binding strigolactones. That is a distinct possibility. But the story could be a little more complicated than that. So maybe Trifasauria's Chi2 proteins can bind strigolactones if those Chi2 proteins are present. But maybe the genes encoding those Chi2 proteins just aren't actively expressed. Maybe Trifasauria's Chi2 proteins can bind strigolactones, but other proteins that are necessary to transmit that signal and turn it into a germination response are missing. So to start to address this question, my student Christy Moss in the right corner here is doing the same thing I did with the Chi2 genes from other parasitic plants in graduate school. She's putting Trifasauria Chi2 genes into Arabidopsis to see if they give Arabidopsis a positive germination response to strigolactones. So when we put a Trifasauria Chi2 gene into Arabidopsis, we make sure it gets expressed during seed germination. So those Trifasauria Chi2 proteins will indeed be present. And we know that in Arabidopsis, the other genes and proteins that are necessary for transducing the strigolactone signal are present. So Christie's experiment will help us determine why Trifasauria doesn't have a positive germination response to strigolactones. Again, do those Trifasauria Chi2 really not perceive strigolactones, or is there something else like lack of expression or lack of another important gene in Trifasauria? The remaining questions don't have any answers yet, but I will introduce them briefly. This is one that I'm really excited about. It involves two local facultative hemiparasite species, and you might be familiar with one or both of these because they do occur in Georgia. So these closely related species belong to the genera Agellinus, that's the purple one here on the left, and Areolaria, the yellow one on the right. And even though they are closely related, they use very different hosts. So Agellinus uses a variety of non-tree hosts, but Areolaria likes oaks. You saw it before in Rachel's host preference experiment. So we can buy seeds for both of these species. We can find them in the field. And my collaborator, Dr. John Stanga from Mercer University, and I have discussed sequencing the transcriptomes of these two species. And what that means is that we'll get all of the RNA, which represents genes being expressed by these two species at a specific point in development. Ideally, the point when the parasite seeks and finds its hosts. And then what we could do is compare the transcripts from each species to see if there are any differences in gene expression between the two that help to explain its very different host ranges. Again, Agellinus on non-tree hosts and Areolaria on trees. Another big question about parasitic plants is how their photosynthesis-related genes have evolved. So remember, some parasitic plants don't photosynthesize, some do, but still require a host. So have any of these parasites lost genes that other plants use for photosynthesis? This project is something that students in my undergraduate genetics class carry out. So they start with the sequence of a known photosynthesis related gene from the model Arabidopsis. And they use a tool called BLAST to search for sequences of the same gene in other species, including parasitic plants. So we take that Arabidopsis sequence, we used BLAST to find orthologs, the same gene in other species, including parasites. And then the students use a variety of different tools to study the evolution of that gene in parasites and in non-parasites. And they can determine if that photosynthesis related gene has evolved in strange or interesting ways in parasites. Ultimately, they write a formal lab report on the project and they contribute to a presentation that is delivered at our college's research day. And this project is actually headed up by Dr. C.J. Tsai, who is a professor at the University of Georgia. And finally, some non-parasitic plants have extra Chi2 copies that have evolved kind of similarly to the strigolactone perceiving Chi2 genes in parasitic plants. So what are those extra Chi2 genes doing in non-parasites? This is an illustration of cassava, and it has 5 or 6 different copies of the Chi2 gene, which is unusual for a non-parasitic plant. So my students, Michael Crow and Madison Hollis, right here, were working to put cassava's Chi2 genes into the model plant Arabidopsis to see if those cassava Chi2 genes give Arabidopsis any interesting new traits. So in conclusion, at this point, we know that some obligate parasitic weeds use extra copies of Chi2 to perceive host-derived strigolactones and to germinate in response, but lots of questions are left to address, and they fall into the fields of genetics, ecology, and even pedagogy, the method and practice of teaching. Now, I might have given you a bad impression of parasitic plants, talking about how destructive some of them are to agriculture, so let me leave you with this. There are lots of parasitic plants that you can find here in Georgia, and most of them are valuable members of local ecosystems. So take a look at these photos and ask yourself if you recognize any of these parasites. So beech drops and Canophilus americana, American cancer root, both lack chlorophyll and they both latch onto the roots of trees. Beech trees for beech drops, oaks for Canophilus americana. False foxglove species belong to Agellinus and Areolaria, and they are beautiful wildflowers that show up in the summer or fall here in Georgia. We talked about mistletoe. It stays green up in the treetops year round here in Georgia, and it's really easy to see when the trees have lost their leaves, but you still have these clusters of green up in the branches. This one right here is called dodder. It also lacks chlorophyll, and it's really interesting because it's a stem parasite. So it attaches to its hosts above ground, and it kind of looks like spaghetti noodles spread out across host plants. And then over here, we have pine sap and Indian pipe, and those are both mycoheterotrophs. They lack chlorophyll, and they're stealing resources from a fungal intermediate. So next time you're out in the woods, see if you see any of these plants. The ones that lack chlorophyll look especially bizarre, so you're unlikely to miss them if they're out there. There's one last thing I want to mention before we get to questions. And this is my newest scientific endeavor, which is only impossible because of training from and collaboration with some amazing scientists, including Dr. Marty Cipollini here at Berry College. So this project is all about restoration of American chestnut trees, and the organization that coordinates this project is the American Chestnut Foundation. So if you haven't checked it out before, I definitely encourage you to go to their website. The American Chestnut Foundation has tons of opportunities for you to get involved with chestnut conservation. You can report chestnut trees that you find growing in the wild. You can volunteer at an event like a chestnut planting, and you can even find some fun social events on there, like special brewery days. And if you don't already know the story on American chestnuts, here it is. American chestnut trees used to be incredibly important species in forests in the eastern US. So the American chestnut species was commercially valuable as well because of its wood and its nuts. So it was really important to forest communities and also for the products that came from it, its wood and the food that you could get from it. So about 100 years ago, an introduced fungal pathogen started wiping out American chestnut trees, and in the wild, they are all but gone. You can still find some that are probably stunted in growth or perhaps resprouted from trees that had been knocked back by this fungus that causes blight. But American chestnut isn't currently fulfilling the ecological role that it once did. And when it got wiped out, the carrying capacity of forest habitats decreased, nutrient cycles were altered. So there's a big push to try to restore American chestnuts and get them back into our forests. So with Marty at Berry and other scientists from the American Chestnut Foundation, we're pursuing two major approaches to chestnut conservation. And the first relies on Chinese chestnuts, which can do just fine with the fungal pathogen, but are not ecologically interchangeable with American chestnuts. So this first approach is a back cross approach. So we cross those rare American chestnuts that are still making pollen with Chinese chestnuts to produce a 50-50 hybrid. These should have some resistance to the fungal pathogen, but they're still not similar enough to American chestnuts to reforest with them. So we keep back crossing those hybrids with American parents until we ideally end up with a tree that is essentially an American chestnut, but has resistance to the fungal pathogen because it's got a few genes from its Chinese chestnut ancestor. Now, this approach is exciting. It's an important part of the program, but it's tricky because resistance to the fungal pathogen probably requires quite a few Chinese chestnut genes, and we don't know what they are yet. So it's really hard to end up with a backcross tree that is entirely American, except for those multiple Chinese chestnut resistance genes. It's a tough approach. So the second approach involves genetically modified American chestnuts. Remember, we don't know what those resistance genes are in Chinese chestnut, but we do know that the fungal pathogen secretes a chemical called oxalic acid. And that oxalic acid is critical for the fungus being able to make American chestnuts sick. Now, wheat has a gene called oxalic acid oxidase, which breaks down oxalic acid. And so with the transgenic American chestnut trees, they are engineered to just carry that oxalic acid oxidase gene from wheat so that they can break down the fungal pathogen's attack compound. The transgenic chestnuts are sort of in their infancy, but I'm really excited to see where they go in the future. So again, if you think chestnuts are cool, look up the American Chestnut Foundation, see what we're all about. I'm still learning the ropes of chestnut research, but as you can probably tell, I'm really excited about it. So with that, I will thank ABG and you for being here. Thanks to Berry College and my lab members, lots of collaborators, including Marty Cipollini, John Stanga, and CJ Tsai, and my organizations, the International Parasitic Plant Society and the American Chestnut Foundation. So with that, I am finished and we can take any questions. So Ian, did you?
Yeah, I didn't see myself there, but I think I must be on. Do you see me? Or can you hear me?
I can see and
Oh, okay, good. All right. Well, thank you so much for such a wonderful presentation. That was really cool. I don't think I'll ever look at another Oriolaria the same. So that was really neat to hear what y'all are up to. And yeah, it'll be interesting to see what comes of some of these interesting projects y'all have going on. But we have lots of questions that have already come in. So if you're ready to get rolling, I can start going with those.
Absolutely. Sounds good.
So we have a question from Bob Stevens, who asked, Chi-12 or Chi-2 gene duplication implies only a second copy of the gene. Are there cases of replication, i.e. more than 2 copies, that could multiply the effect and or neo-functional opportunities?
Yeah, great question. So there are definitely successive duplications that have happened in the parasite lineage. So parasitic plants, some of them only have a single Chi2 gene copy that appears to be pretty rare. Most of them have many more. One species has up to, I believe, 21 copies of the gene. So yeah, it has been duplicated pretty extensively. Over and over again. And many other parasite species, including lots of parasitic weeds, have extra copies, maybe in the range of 4 to 8, 4 to 12, something like that. So yeah, lots of copies.
Okay, we have a question from Jordan Argret, who says, hey, Dr. Conn, I am an incoming UGA graduate student looking into hemiparasite impacts on litter dynamics and local microbial community. Do you see native on weedy parasitic plants as a potential mechanism for conservation/restoration work due to their positive impacts on species diversity due to host suppression? Slightly different topic, but I thought I'd ask. Big fan.
Yeah, that is a fantastic question. And I think you're headed to a fantastic place. UGA is an awesome school to do plant research. And I'm really interested to hear more about the direction you're going to take with that. I have read in a paper that parasitic plants can be useful in that way, and that, they can parasitize maybe a dominant member of an ecosystem and knock it back a little bit so that other species have a better chance at growing. So I can't remember the paper where I read that, but that is sometimes something that I mention in my little shout out to parasitic plants at the end of the talk, that they can be actually really good for the diversity of ecosystems in that regard. Great question, and have a great time with grad school.
We have a question from Ethan McQuahe, who asks, could the research on Ki2 genes added to other host plants be applied in agriculture? With near 90 to 100% germination rates with artificial SL, that seems like a no-brainer in application. What would you see the consequences of what that would be?
Yeah, so that is a really interesting question. I guess first you would want to apply this to maybe something that you couldn't get to germinate very successfully on its own, because it wouldn't be the easiest thing in the world to get those Ki2 genes into crop plants and then, get it widespread enough in a big enough population of the crop plants that you can spread them out across a large geographic area and grow them. So if you had a crop that was really reluctant to germinate, then it could be a way to go about that. The other problem with using strigolactones is that they are relatively unstable when they are applied artificially, and they're also pretty expensive to produce. So when we're talking about controlling parasite infestations in fields, one option for getting rid of the parasite infestation is just to treat the whole field with strigolactones. They'll germinate, you don't have host plants for them, and they'll die pretty quickly, right? Because there's nobody there for them to latch onto. But the problem with that approach is that the strigolactones are expensive and are unstable. And so it's probably not the most practical approach in that regard. But you're right. I mean, it's a high germination rate.
That's interesting, though, that people have tried that. It's too bad it's cost prohibitive.
It's just not great for large scale application.
Cool. All right. So we have a question from Patrick Ardif, who asks, was there any difference in the growth of the host when the parasite plant was grown with it?
That is a great question, and is something that we are definitely going to look at in the future. The experiment that we did this past spring ended up just being something we kind of threw together. I got the oak seedlings from my yard. They were going to get mowed over anyway, so I just dug them up and brought them into the lab. So I think that is a fantastic question. And it's an easy one to address because they're just seedlings at this point. So when they're full grown trees out in the wild, it's kind of hard to tell if parasites are impacting them. But I think that is definitely something that we can and should address in the lab in the future. Great question.
Okay, and a question from anonymous attendee who asked, I once saw a common milkweed plant that was completely white. I was told that since it was white, it lacked chlorophyll, and so it was behaving as a parasite. Are you familiar with plants that are typically not parasitic behaving as parasites? Is this found more often in milkweed plants?
I am not at all familiar with that. I have not heard of that before. There are definitely mutant plants out there that aren't parasites, that aren't making chlorophyll for whatever reason. If you take a plant and deny it light for a while, it'll start to break down its chlorophyll and not produce more. But No, I have never heard of that. It's possible that if it lacked its chlorophyll, it was surviving just, on sugars that were stored in its roots. But I don't know of any way that a plant that isn't parasitic would be able to grow a hostorium and latch onto a host. That is very interesting, though. I'll have to do some digging
Yeah.
about that case.
Yeah, I haven't heard of that with milkweeds, but yeah,
Yeah,
interesting question.
it's
Thank
definitely.
you. And do we have any other questions from anyone? I think well, feel free, folks, to put any more in the Q&A section if anybody does have any questions. I've got a question for you. I'm just curious, do you have a particular like favorite parasitic plant that you've worked with over the years?
Yeah, well, yes. I guess I have a few favorites. I mean, in grad school, I didn't work with the parasitic weeds, just with their DNA. So they were really exciting to work on because of the real world applications. And grad school was just a good time. So I guess that I reflect fondly on my work with those weeds. I wouldn't say they're my favorite because they're very destructive, but I think Canophilus americana is probably my favorite. It's just so exciting to see it in the forest, and it looks so strange with no chlorophyll and its small stature. I think people do mistake it for pine cones sometimes, and its distribution just seems to be really patchy. You'll walk for miles and miles and miles through the forest, and suddenly there'll be a little patch of it, and then you won't see any more of it. So it's kind of mysterious and beautiful, but also very strange looking and just a fun one to see.
It's associated with oaks too, right?
Yes,
Yeah.
yes,
Okay.
it
Thought
is.
so.
Yeah.
Yeah.
Yeah. And as far as I can tell, nobody knows how to make it germinate.
Oh, really?
Yeah, which is
Wow.
which is pretty
Yeah.
cool. The obligate holoparasites are tricky to make germinate, which is understandable because if they germinate at the wrong time when there's not a host there, they're in big trouble.
So, another question about Conopheles? How much do you think? Like, I've always heard that bear, they call it bear corn, and people say
Yeah.
bear like to eat it. Do you think they do that much? I've always wanted, like, because I see it and I'm like, I don't see this bear corn that's just getting devoured all over the place, but maybe they eat the whole thing, and I just don't.
That's a great question. I
Yeah.
don't know. I have
I,
never personally encountered a bear eating it,
Yeah,
but
I haven't either. I've just always been curious if that's just kind of like something that people say, or if it's something that, I've heard in the springtime, there's not much to eat. They'll eat a little
yeah,
bear corn,
yeah, I don't know.
kind
And
of
it is
clean
really,
out the pipes a little bit. Yeah.
yeah. And maybe that's why its distribution is so patchy. The bear eats it and walks a long way and
Yeah.
drops the seeds. And yeah, I don't know. That's a great question.
Oh, I think I see another question in the Q&A.
Oh, yes.
Oh, we got a
Okay.
few more.
Oh,
Yeah.
wow.
Okay. So at one point, I had heard that this is from an anonymous attendee. At one point, I had heard that parasitic plants had no evolutionary significance. I feel that they do. What is your view on this?
Yeah. I mean, I would like to ask the person that told you that what they were thinking because I absolutely think they do. I mean, I think it's just fascinating when species evolve with one another and adapt to one another. I mean, parasitic plants are just this amazing case of diversification. In the Orobanchaceae, they are so diverse, not only in terms of their host dependence, but in terms of what hosts they use, how many different hosts they can use. I think a really fascinating thing about them evolutionarily is that so many of them have evolved to use crops as hosts. And in the grand scheme of evolution, agriculture is not that old. So they've adapted to use crops as hosts. They have this amazing evolutionary adaptation adaptation that makes sure they don't germinate when a host is absent. Presumably, their host plants have evolved in response to them as well. And so, yeah, I think they absolutely have evolutionary significance. And I think that the more we can learn about these amazing cases of adaptation, the more we understand the world around us. And another really cool thing, I think, is that in a lot of cases, parasitic plants that are dependent upon a host have lost things that other plants usually have. So a lot of the obligate holoparasites have lost chlorophyll. They don't need it anymore. They're not making it. A lot of them basically don't have roots or leaves. They might have really basic or small roots and leaves, but they don't need those structures anymore. And so in a lot of cases, they've all but lost them. In some cases, they've lost a lot of their genes. And so this theme of losing things, it happens a lot in parasites that have a host that they can rely on that's fulfilling those needs. So I think that's a really cool evolutionary aspect of these species as well.
Okay, we have another question from another anonymous attendee who says, I've understood that white pine blister rust jumps from one host to another in ribes and pinus. Do other parasites do this?
Yes, absolutely. So host range and, host compatibility is something that I think is so interesting for parasites and pathogens. So, anytime a new disease breaks out, for example, we've seen it with COVID-19, we've seen it with HIV, we've seen it with Ebola, lots of other diseases, avian flu, breaking out among humans. Whenever those kinds of things happen, it's usually because there's a pathogen out there, which is essentially a type of parasite that used to use one particular group of hosts and suddenly, for whatever reason, has expanded its host range and is now able to jump into a new host species. And exactly how that happens is something that isn't totally clear, but genetic mutations that end up getting passed on almost certainly facilitate it in a lot of cases. We see it with parasitic plants too that have adapted to crop hosts. In a lot of cases, you can track the history of that species and see that it used to parasitize wild hosts. Sometimes populations of it still parasitize wild hosts, but populations of it have also jumped into agricultural hosts. And we can see it with fungal pathogens, all types of pathogens and parasites. It's such an interesting phenomenon.
And one last question from Caro Frost. I wonder if the plants that parasitize herbaceous plants are more common on grasses that use C4 in hot, dry climates.
That is another great question. Great questions tonight. So I don't know totally what the connection is between climate and the parasites in that area and what hosts they specialize on, but definitely the Striga species, the witchweed species, do use grasses as their hosts. So things like maize and rice and sorghum are species that they parasitize, and whether they're parasitizing them because those plants, because of the photosynthetic mechanism of those plants, or whether they're parasitizing them because they occur in an environment where C4 plants just happen to be doing the best. I don't know, but yeah, there are definitely parasitic plants that specialize on grasses.
All right. And this will be our last question of the night. It's from an anonymous attendee who asks, have you considered working with the American beech and the parasitic beech drops and their impact on the growth of the American beech tree?
Yeah, so I haven't thought, trees are pretty new to me. So I don't have a way in mind to look at the growth of beech trees and how beech crops might impact them, other than, possibly having the seedlings in the lab and measuring the growth of seedlings. So when it comes to like a big tall tree in the forest, I don't have the training to necessarily know how to work with it, but I think that is also a great question. Beech drops would be really convenient to work on, at least for me, in that I tend to see them more often than I tend to see other parasitic plants. And so, yeah, I think that's a fantastic question, because I think it's generally assumed that tree hosts don't suffer too much from these little root parasites, but as far as I can tell, that's an assumption. So it would be a great thing to test.
All right, there's another one for the list.
So many questions,
Okay.
so many research projects to be had.
Okay, well, again, Dr. Conn, thank you so much for being here with us tonight and presenting and answering all these questions. It was wonderful, and I hope it was enjoyable for you.
It was great. These questions were fantastic. I mean, this is probably the best set of questions I've gotten.
That's great. So I'm going to go ahead and just share my screen now.
Yeah.
I thought I was sharing it. There it is. Okay, again, just want to thank everybody for coming out tonight. We've, really appreciate having, these wonderful speakers, and we also appreciate having all of you that come out to be a part of this. So again, if you'd like to hear about more talks that we have coming up in the future, you can check out our Science Cafe webpage. And again, we will go ahead and put a recording of this event. So if you want to share it with any of your friends who weren't able to make it, you'll be able to do that. I'll try to have it up on the website in about a week or so at the latest. Also want to talk about our next webinar, which is going to be Thursday, August 19th at 6 p.m. Eastern Standard Time. And that'll be with Kendall Jackson, who is an archaeologist and historical ecologist and PhD candidate at the University of South Florida, who will be presenting on his recent study on microfossil evidence for Native American wetland plant use on the Gulf Coast. So a little bit of ethnobotany, paleoethnobotany for you there. Again, if y'all have any questions, suggestions, or anything like that about Science Cafe, feel free to reach me at my email address, which is isabo@atlantabg.org. And also just want to share with y'all that we do have an Instagram that is Atlanta BG Conservation. And also, Dr. Conn, do you have, if folks are interested in hearing about some of your projects, do you have a social media? I think you do have a Twitter and.
I do. Do you want me to put it in the chat there?
Yeah, that might be good.
Okay, so
share your, I'm sorry, I should have shared that earlier. Just thought of that.
That is okay. Okay, there's my Twitter handle and my lab website. Oh wait, that just went to the panelists. Hold on. I'm not used to this level of organization on a Zoom.
Yeah.
Fancy.
I'm still learning myself, so.
Okay, there we go.
All right.
Yeah.
Great, thank you. So yeah, folks can check out what Dr. Conn is up to there at her Twitter and also her website, which I've already checked out. It's really cool. You all should too. And again, thanks for everyone who came tonight. I'll go ahead and
thank you, everybody.
Stop the recording. Let's see, stop screen.